issues of our meliponiculture first, and then what are the objectives of current studies, brief method on the in vitro rearing, and then also the results that we found, which include of the growth and ontogenic time of in vitro rearing of the H. itama under lab conditions, and how we manipulate the larva food amount, and also the effect of symbiotic with fungus on the queen of stingless bees. And last but not least, our ongoing field experiments where we have introduced our in vitro queen in the field. So for the introduction, as we know, meliponiculture or rearing of stingless bees in hives currently is a very common activity in Malaysia. And we know it is an emerging new source of income for many Malaysian beekeepers. Why? This is due to the high demand on the stingless bee products, especially the honey, as well as the propolis and pollen. Besides that, we know that stingless bees are very cultural pollination, where I think a lot of papers or scientific reports have reported that this is the crop. And in Malaysia, there are 4 species of have been popularly or commonly domestic, which are Heterotrigona itama, Geniotrigona thoracica, Tetragonella atripus, and Tetragona penicillarius. But out of these 4 species, the H. itama is the most popular domesticated species. So the current issues of our meliponiculture in Malaysia is because of these 4 major problems. So actually there are a lot of problems, but the major problem or bottleneck of meliponiculture in Malaysia, first is the rearing of the queen. We know that the queen is the main component in order to multiply the colony. So if you cannot get the queen, so the colony will be collapsed. And then there are certain areas where the low honey production have been reported, and then the quality inconsistency of the stingless bee honey, as well as the price, which some of the areas or state the price are extremely expensive. So we need to multiply the stingless bee colonies. Why? Can you see my presentation?
No. You have to upload, share again.
Okay. Mass produce of the stingless bee queen. So this technique is considered very simple and very sophisticated, where you can multiply rather than naturally produce queen. So using this technique, we can overcome the low production of the queens. This technique actually focusing on the female larvae, where we can manipulate the larva of food as well as the symbiotic bee to make sure that. So far have been used this technique, such as the Skeptotrigona plebia dry and also the Friso melita, but none of this is actually our native. So we want to try technique with our native species, which is H. itama. So this is the basic cycle of stingless. If you can, so I actually refer to Salma, where their group have already reported about of H. itama, but this is based on the field. So from their report, they found that they need 46.5 days to develop from eggs until the adult stage. But here, if you look here in this picture, this is the queen, where the queen will the egg brood cell, and then the eggs will hatch within 2 to 5 days. So during this period, as you can see here, the larvae will eat the food, which actually in liquid form inside the brood cell. So the larvae will eat around 10 to 15 days until it finish, and it will stop eating when it reach to the pupa stage. So for pupa stage, it takes about 1 month, and it will transform slowly to be adults. So this is what happening inside the hive of the stinging bees. And the finding, there's a finding where they found white fungus grows from the border of the brood cell of the larva food. So this fungus is very important fungus, where this fungus need to be used during the larva development. It starts proliferate after 3 days after eggs being laid, and we also found this observation experiment. Okay, so study our which is Heterotragonite tama, because as I said, this is the most popular species domesticated in Malaysia. And besides that, they have their unique characteristic, where they are polylacty, adaptability, floral fidelity, easy to be domesticated, have permanent colonies, have large food reserve, and a very good pollinators for many crop of species. Yeah. So in this study, I would like to share. First of all, I determined. Me and my team, we determined the growth and development of these species under in vitro conditions. After we understand the requirement of the humidity, the amount of the larva food, and so on, we want to develop. We want to read the queen. With this specific objective, we manipulate the food amounts of the larva in order to produce the queen. And last but not least, we want to investigate the effect of the bee fungus on the survivorship of the larvae of H. itama via in vitro rearing. So the samples, we collected the samples around Terengganu area, where we only took one or two combs of the brood in one colony in order to avoid the excessive damage. And I think we have taken more than 500 brood cells so far for this study. And what we did, we gently squeezed the brood cells to get the larva food, where we want to we investigate the microbes from the larva food. And then we gently take out the eggs from the brood eggs, and then we put into the ELISA plate, and then we rear inside the incubator. So that's a very brief method. And then we incubate the larvae inside the incubator using certain temperature and also humidity. And then we measured the survivorship, the growth, the larvae, until it turned into the adult stage. And then we analyze using the statistic. So that one is for the development or the growth of the species inside lab conditions. Now, in order to produce the queen, we have to manipulate the larva food. So there are 3 types of treatments where we manipulate the amount of the larva food. But before we decided to put this specific amount, we have actually tested. We have already measured from the field the amount of the larva food inside the H. itama workers cells. So from these, we have do the trial where 3 times, 4 times produce, because the queen cells or maybe 5 times larger from the worker. That's why we have the larva food is higher from workers. So we tested using these treatments, and then these are the relative humidities that we control and the temperature. For the third objectives, where we want to test whether the fungus is very important during the growth stage of the H. itama, we have 3 treatments. First treatment, we autoclave the larva food, meaning that we kill all the microbes inside the larva food. This acts as the control treatment. And then treatment B is the fresh larva food taken from the field. And last, treatment C, the autoclave food, plus with the pure culture of the symbiotic bee fungus. But before that, we do the pure culture, or we mass culture the symbiotic bee fungus first. And before the experiment, we isolated and identified first the symbiotic bee fungus, where we followed the Paludo et al. Methods, where we isolate the fungus and we mass culture inside the selective media based on the method of the Paludo, and then we do the identification based on microscopic as well as molecular analysis to identify what is the species of the fungus. Okay, now we go to the results and the discussion. So this is actually the growth of the H. itama under in vitro rearing. As you can see here, from our observation, we found that the H. itama needs 6 larva instar stages in order to develop from eggs to the larvae to the pupa and then adults. So these pictures showing the rearing, the growth development of H. itama. As you can see here, the eggs need 1 to 3 days to hatch. And then for first instar, it will need 2 to 3 days. And then after that, they will molt the skin. They will go to second instar, fourth instar, fifth instar, and sixth instar, about 20 to 20 days. Okay, and then they will move to the pupa stage where they will stop eating, and they will be around 20 to 50 days during the pupa stage, and then they will emerge to the adult. So this is basically the ontogenic time of these species. Oh, sorry. And we measure the measurement of the head capsule, and it increased from time to time. And if you look here, the queen and the worker is significantly different. I mean, during the week 5, yes, you can see here the size of the queen is 2 times larger than the worker, even though they are in the same stage. So these are the basic ontogenic time. So it is actually similar to the finding by Prof. Salma, where we also found the species need 46 to 50 days to develop from eggs until to adult stage. Okay. All right, now we move to the next objective, where we manipulate the food amount, the larva food amount. So in this study, if you can see here, for the first treatment, we got a lot of worker females, where 78% of the treatments have been merged to the worker adults for the 100 micromil of larva food. But for the 120 micromil, a lot of mortality have been observed. But still, the worker can be produced. But the amazing one is we have 40% of the treatments have been emerged into a queen. And last but not least, the last treatment, 150 micromil, 78% of the eggs that we treated have been emerged to the queen. So these experiment have showed that the larvae treated with the high amount of the larva food have emerged to the queen. And then if you look here, from these, we can suggest that in order to produce the queen, at least 120 to 150 micromil is needed to make sure that the larvae can be developed into the queen. Okay. And before that, we will publish this paper at the end of this year, and this paper has already been accepted by one of the journal. And for the next objective, where we want to understand the effect of the bee fungus, we have do the experiment based on 3 treatments. As you can see here, the survivorship, if we autoclave the food, the survivorship is very, very low. Only 37% will survive. But the survivorship is not until the adult stage. Yeah. But in contrast, the treatment B, fresh larva food, more than 80% have been survived until the adult stage. And the treatment C, where we autoclave the food and we mix with the pure culture of the symbiotic fungus, only 50% were survived. So it shows that the bee needs the larva food, the fresh larva food, because the fresh squeezed larva food probably contain other beneficial microorganisms plus with the symbiotic bee fungus, which need to be further explored. So I just want to show you the pictures of our experiment. If you look here, the treatment A, where we autoclave the larva food, more than half of the treatments cannot be further developed, where you can see here the eggs were not viable. But for the freshly larva food, the fungus have been developed until the adult stage. And if you look here, the fungus appear on the border of the brood cell. So it shows that there are a lot of beneficial microbes inside the larva food that is required for the larvae. And then we isolate the fungus. We want to identify what are the species of the fungus. So based on the morphology, it shows that this fungus belong to the genus of Zygosaccharomyces. But under genus Saccharomyces, and then we further investigate with the molecular. So from the molecular, it shows that the B fungus band appeared in the range of 3,000 to 1,000 BP, which where we sent to the company to verify the species. It shows that the species was actually identified as Candida species. So the Candida species is actually a group of fungi under the order of Saccharomyces and so it is also have been supported by previous reports from other researchers, I think Paludo et al., if I'm not mistaken. So it shows that the species fungus from the Brazil. Okay, just want to show you the video that we took during our in vitro rearing. So if you can see here, this is the pupa of the H. itama inside the incubator. Here, yeah, you look here, this is the larvae. So this is the mouse part of the larvae. You can see here, the larvae actually is consume the fungi that is that can be found around the of the ELISA plate. So the larvae, they eat 24 hours during their stage. So if we remove the fungus, the larvae cannot be survived. So the fungus is very important during the larva stage. So after the adults, the larvae, and then they turn to the pupae, and then finally this is our outcome where we really produce the queen. And for information, we have produced more than 50 individuals of the queen so far from our experiments. And then in order to verify whether the queen that we produce is similar to the natural queen, we have to measure the morphology as well as the reproductive system. So what we did, we measured the size, and it shows that the in vitro queen is greater than the worker female. And also now we are actually comparing with the natural queen. It's very hard to get natural queen because we need to get from the field, and it's very hard to get the from the beekeepers because they don't want to sacrifice their queen. So that's why in this study I only compare the queen of in vitro with the workers. But the comparison with natural queen is actually under is ongoing, yeah. And then okay, and after we measured the morphology, we dissect the abdomen. From the abdomen, we want to see whether the in vitro queen have a very good reproductive organ. So if you look here from the figure A, this is the reproductive system of our in vitro queen, where you can see here the ovariole. So this is the organ where the eggs were produced. The ovarioles are well developed, and compared with the B. B is actually the reproductive system from the female worker. So female worker, they don't have well-developed ovarioles, okay? And then one of the most important thing is the spermatheca. So we know that spermatheca is a very important organ. When the queen mates with the male, they will put the sperm inside the spermatheca. So spermatheca only have inside the reproductive organ from the queen of in vitro, but not from the female worker. Okay, after that, yeah, we finally produced. So the most important part is whether our queen is fertile or whether they can be accepted by the the wild colonies in the field. So if you look here, yes, we have produced a lot of queen. Now it's time to produce in the field where we have introduced to the newly queenless colonies. So I just want to show you, this is the pictures that have been observed after a few weeks. I think it's more than 1 month, where it shows a very positive result. But honestly, before we success or we found a good positive, we actually face a lot of failures because we have to understand the behavior. So for the first time, if you look at the red circle, the workers kind of rejected the queen, where they killed the queen. They mummified. They put the propolis on top of the queen, where they kind of prison the queen. Even though the colony doesn't have any queen, but I don't know, maybe they cannot accept the new queen from, not from the natural, but from the artificially produced queen. But then we try to modify our technique, where when we want to introduce the queen, we put the queen inside you see here the queen cell. So the queen cage, the queen cage is actually from the Apis queen cage, where we put our queen inside the cage, and then we put inside the colony that have a few broodies, a few propolis, and alhamdulillah, after a few weeks, the workers accept our queen. So we finally opened the queen cage, and this is our queen. So we take out our queen and then put it inside the new colony, and now it is under our observation to see whether the queen can produce new generations of the brood cells, and we need to test whether the eggs produced by our in vitro is viable, is fertile or not. So this is under ongoing experiments. So this is another replicate of our study. Okay, so you look here. So the queen actually need to be put inside the queen cage first. So that is the technique that we have improved. So if you look here, this is the new colony that queenless. So if you look here, the workers from other colonies have come into the new colony, means that they have accept our queen, right? So now we are trying to observe whether the propolis has been developed, have been increased. So it shows that the workers from the field is accept of our queen. Okay, so all right, now we go to the last slide, the conclusion. So from our study, we finally come out with the development time or the ontogenic time of the H. utama, where it shows that H. utama need 46 to 50 days to develop from eggs, larvae, pupa to adults, and there are 6 instar stages ranging from 0.64 mm to 1.99 mm, the larva stage size. And then from our manipulation of the larva food, it shows that the larvae needs a very high amount of larvae food, which is around 120 to 150 micromil in order to produce the queen. And then we have verified that the queen that we produce have a very huge size with huge abdomen with well-developed reproductive system. And from the in vitro rearing, we found that the larvae crucially really need the beneficial fungus in order to pupate. If we the larvae will not develop until pupa stage, and the higher percentage of survived larvae can be found from the food that supplemented with the fungus. And now we have very good positive findings from the field trial, but ongoing experiments where we need to verify the fertility, the fecundity, kesuburan of the queen, and then if the queen managed to produce eggs, we need to check whether the eggs will hatch, become the adults or not. So it's still ongoing process, but it shows a very positive result from the field.